Now let's learn about steps in rDNA technology. So there are various steps. We have already had an introduction about the steps in rDNA technology. Now we will study one by one. First is isolation of DNA. What's the meaning of isolation of DNA? We need a foreign DNA, a desired gene. So that gene should be isolated from the cell of another organism. That is a donor. So usually the donor can be a bacteria, or it can be a fungi, or it can be a plant cell, or sometimes it will be an animal cell. So if we are going to take it from a bacterial cell, first the cell wall should be digested, removed. For that we use the enzyme lysozyme. If we are isolating the DNA from a fungal cell, we use the enzyme chitinase, which can digest the cell wall chitin. Chitin makes cell wall. Now, if it is a plant cell, we use the enzyme cellulase, which can digest the cellulosic cell wall. The cell wall is digested. Now, when the cell wall is digested, we will get the inner parts. In that, we will get the nucleus. Then inside the nucleus, we will get the DNA. Or DNA will be covered or bound around by different types of proteins, RNA and all. We need to separate all these things. So to remove proteins from that, we use the enzyme protease. Then to remove RNA from that, we use the enzyme RNA. Some other chemicals are also used to remove the membranes and all. So these are the things which we should know. That is the enzyme used to remove the cell covering, cell wall coverings, and also the proteins and RNA bound to the DNA. Now we have cleaned out this DNA. Now we will precipitate the DNA. That is, the purified DNA is precipitated as white threads using chilled ethanol. തണുപ്പിച്ച ethanol അതിലേക്ക് ഒഴിക്കുമ്പോൾ DNA white threads ആയിട്ട് കിട്ടും. ഇത് നമുക്ക് പ്രാക്ടിക്കലി ചെയ്യാനായിട്ടുള്ള ഒരു എക്സ്പെരിമെന്റ് ആണ്. To precipitate out extraction of DNA എന്ന് പറയുന്നത്. So അത്രേ ഉള്ളൂ. So that is the first step, that is isolation of DNA. Now the second step is fragmentation of DNA. We don't need that entire DNA. We need only the portion that is with the desired gene. For that we are going to fragment or cut the DNA into fragments. For that we use the enzyme restriction enzyme. Here the restriction enzymes allam the optimum temperature, pH, all these things are very important. So we will incubate, means along with the DNA, the restriction enzyme and the suitable temperature, everything is given, so that the restriction enzyme will act on it and will cut into pieces. Now we are having DNA fragments with us. Now we have to separate the DNA and we need only the desired gene. So we need to sort out the desired gene. For that, for separating the desired gene, we use a method called as gel electrophoresis. Okay, this is an equipment which can be connected to an electric field, and it's having a matrix. And this matrix is made up of agarose gel. Agarose gel is a gel obtained from the seaweed. It's a natural polymer obtained from seaweeds. കടലിലുള്ള large algae നിന്ന് obtain ചെയ്യുന്ന gel. And we are using this because this agarose gel is having a natural sieving effect when it is connected to the electric field. അതായത് ഇങ്ങനെ അരിക്കുന്ന പോലെ അതിനെ സൈസ് അനുസരിച്ച് മൂവ് ചെയ്യാനുള്ള ഒരു കഴിവുണ്ട്. That's why we are using it. Now, so this full thing is connected to the electric field. Now, the DNA fragments will resolve. Resolve means will separate according to the size. It means larger one will move only slowly, whereas smaller one will move faster. അതായത് ഇങ്ങനെ ഇരിക്കുന്ന ഒരു സ്ട്രക്ചറില്. So if this is the gel electrophoresis, that structure, that machine, then here there are regions called as pits or wells. Wells are ഈ വെൽസിലേക്കാണ് നമ്മൾ DNA fragments ഒഴിച്ചു കൊടുക്കുന്നത്. Pits പോലെ ഉള്ള regions. അതിന് നമ്മൾ fragments ഒഴിച്ചിട്ട് when we connect it to the electric field, what will happen? This DNA is negatively charged. So it will start moving towards the anode. Okay. So now from this, it will move according to the size. So the larger ones will move. So the larger ones will move at a faster rate, whereas it's connected to the electric field. So the larger ones will move slowly, whereas the smaller ones will move faster towards the anode. Anode is positive charge end. So since DNA is negatively charged, it will move to the positive end. Now here the question is, So smaller fragments can be seen where near towards the end, whereas larger fragments will be seen near to the wells itself. It will not move that much fast. Now, what makes the DNA fragments move? Two things. One is when it is connected to the electric field, DNA being negatively charged, it will move towards the positive end. Second thing is that the sieving effect of the agarose gel also helps the separation of the DNA fragments. So now the DNA fragments are separated like this. Some are here, some are coming to this end like this. It will get separated. Now it is stained using a chemical called as ethidium bromide. That's very important. Ethidium bromide is carcinogenic. It can cause cancer. So it's not safe to use it frequently. So anyway, it is stained using ethidium bromide. Then it is visualized using UV radiation. Stain ചെയ്തിട്ട് UV radiation അതിലേക്ക് അടിക്കുമ്പോൾ we can see it as orange bands. DNA ഉള്ള portion ഒക്കെ ഇങ്ങനെ orange bands ആയിട്ട് കാണാം. So now we have sorted out and we are seeing it as orange bands. So there are methods by which we can separate the DNA of interest. For that, ബാക്കി നമുക്ക് ഇവിടെ ഇത്രയുമേ നമ്മൾ ഇവിടെ പറയുന്നുള്ളൂ. There are some other methods also, but we are not mentioning it. Now from this, the desired DNA is cut out from the gel and we add certain chemicals so that the DNA will get separated from the gel. And that process is called as elution. So by the process of elution, what will happen? The DNA along with the gel is cut out. Then the gel is dissolved out using certain chemicals, and that process of separating the DNA from the gel is called as elution. So only that much will come under separation of gene of interest. So it is done using the method or process called as gel electrophoresis. That's all.